Well, thank you, Boo, very much. And Bengt, happy birthday. Is this, are these your initials? Good. Because I thought they were, but I wasn't sure. So happy birthday. I'm very pleased to be here to give you this talk on our work on electron flow through proteins. Let me start by saying, although there were lots of good reactions yesterday, these two reactions are much more important. In fact, these are the two most important reactions on our planet. The oxidation of water in photosynthesis to oxygen to liberate protons and electrons, the fuel that we need for life. And the reaction which oxygen comes out and we breathe it in and store energy in our bodies for life and respiration, the re-reduction of this oxygen in the enzyme cytochrome oxidase in our mitochondrial respiratory system. The 4 protons and 4 electrons come back to give water. These reactions work almost perfectly. These 4 proton reactions, you always get oxygen here rather than reactive oxygen species. Here you always get water instead of reactive oxygen species. If these reactions weren't running so efficiently, we wouldn't live very long because we would die from reactive oxygen species, which would make us age very, very quickly. So I've been interested in how these reactions work for many years, and I'm going to tell you the stories today about how we understand electron flow through respiration and through photosynthesis. To start, in respiration, when we started this work many years ago, believe it or not, Roger, we didn't know the structure of cytochrome oxidase. We had cartoons. We knew they were big molecules. We knew the metal ions involved in the electron transfer reactions were in very low concentration, so we knew they had to be very far apart. So the question was, how do the electrons really move through these big machines at high speed? Because we know the turnover in mitochondrial respiration is the order of milliseconds. Oxygen is reduced to water of the order of milliseconds. And these are very, very long distances. How in the world do the electrons get through here so fast? They must go much faster than milliseconds. And secondly, they always go to the right places. How can they figure out, they're wandering through these, how can they figure out, there must be some communication system that we have to discover. And coming from inorganic chemistry, I was particularly puzzled by this because in inorganic chemistry, we knew that reactions in water between metal ions were electron transfer reactions were very slow because the barriers were very, very high. For example, from reactions that were done, so-called self-exchange reactions in water, we knew that in contact, because of the high barriers, the water solvation shell was very asymmetric, had to reorganize to a symmetric shell in order for an electron to flow between iron 2 and iron 3. This very high barrier of the reorganization of water around a metal ion meant that the tunneling time from iron 2 to iron 3 was the order of 100 milliseconds. It was already in contact 100 times slower than we knew in mitochondrial respiration. And I dramatized this by putting them 20 angstroms apart, which is what we guessed they were in cytochrome oxidase. And calculated with a square barrier model, I calculated it would take 10 to the 16th years. Bengt, and we don't have that kind of time, Bengt. I know you're 70, but we can't wait that long to, make one water molecule. And so something's going on here in biological electron flow. And what's going on, of course, are proteins that And I proposed many years ago that the protein would fold around the metal sites. There might be cofactors, iron, sulfur, porphyrins, and so forth, but the protein would fold around these redox sites and reduce the barrier by squeezing out the water being the culprit because of reorganization, and embedding these redox sites in hydrophobic and very low dielectric. You would in fact drop the barrier for reorganization, because now there's nothing to reorganize in a hydrophobic cavity. Drop it tremendously, and so to the point where electrons could flow out very rapidly. And then, of course, how do they know where to go? What is the guidance system? Of course, I didn't know what it was many years ago, so I put in squiggly lines. Say there must be some guidance system, which we've now found out after years of work is a quantum mechanical effect. It's quantum mechanical superexchange, distant electronic coupling between these two sites. It allows the electron here to spot this site and come over here rather than something else. So, Bengt, if you want to go to sleep now and sleep through this talk, I'm just going to put some numbers on these two concepts of barrier lowering in the protein cavity and superexchange coupling, so I can wake you up at the end and tell you some new stuff that we're publishing in QRB Discovery. So we're off and running. We invented to interrogate this model I developed many years ago. We didn't need an experiment. And the experiment that we developed at Caltech was a laser flash quench experiment. It goes like this. You covalently couple a probe, a redox accessible probe, covalently coupled to the surface of a structurally characterized protein, such as cytochrome C or other proteins. You couple it covalently here at a fixed distance, 18 angstroms in this case, from the iron center. So you have a fixed distance experiment, so you can see if an electron can in fact tunnel over this distance at some reasonable rate. You have a laser pulse that excites the ruthenium complex, the probe, to an excited state, which is a very powerful reducing agent. And so a quencher in solution can remove the electron from this site very, very quickly, exposing a ruthenium 3+. The electron then in the iron center spots the laser-triggered ruthenium 3+ and tunnels. I'll now start using the word tunnels. Tunnels through this 18 angstroms, and we measured that tunneling time as 500 nanoseconds. It's not 10 to the 16th years. In a protein, we can do it in 500 nanoseconds, which is, of course, plenty of time for mitochondrial respiration. So what we did at this point was to appeal to the semi-classical electron transfer model, which is comprised of this Marcus activation classical part and the quantum mechanical part developed by many people, the so-called electronic coupling part. So this is the barrier part, and this is the quantum mechanical superexchange part, the guidance system part. So we decided experimentally that we would experimentally factor this semi-classical expression by doing experiments with our probe, so we could go through driving force space and find the activationless point in this reaction. After we go through driving force space and map this out experimentally, because we can do that by varying the potentials of our probes, we find the activationless point, at which point this lambda, this reorganization is experimentally determined. And on top of that, this so-called Frank-Condon factor becomes 1. So at this point, the reaction is entirely controlled by the guidance system, by the electronic coupling. So we decided to go through free energy space, check it with temperature, to go down in temperature to 77 Kelvin to make sure it was activationless. So these are the experiments that we did over many, many years to map this, this tunneling out. And the results The results are here. What we found is that, of course, in water, the barrier is very high. The reorganization barrier is 2.7 electron volts, which means the barrier is very, very high, as I pointed out. But in proteins, in every one we mapped through free energy space and temperature space, we found that the barriers are very, very low, so low that the thermal barrier is really low enough that you can tunnel through here at room temperature and and so you don't blow up, you don't overheat. You can run cool with these low barrier reactions for electron flow. So now, having mapped that out, we're now left with the guidance system. What in the world is the guidance system? Now that we know the protein allows the barriers to drop and the electrons to come out rapidly, what's the guidance system? In order to map that out, we decided that we would make a series of electron tunneling mutants at fixed distances, because the theoreticians had proposed to us that the guidance system was quantum mechanical superexchange. And superexchange is, due to overlap of orbitals and so forth, is predicted to be exponentially dependent with distance. That means the tunneling rate would be exponential with distance, and so we wanted to check that out experimentally. In order to do that, I had Ralph Langen in my lab make 6 tunneling mutants of the blue copper protein azurin, where we could make mutants and put our probes in these crystallographically determined tunneling positions from tunneling distances from metal to metal, less than 16 angstroms out close to 30 angstroms, enough tunneling distances that we could test whether the guidance system was really an exponential one or not. Then we went through a series of flash quench experiments through temperature space and free energy space to find the activationist point. We checked it in some single crystals at low temperature. We did literally hundreds of experiments and ended up with a tunneling timetable, which I show here. These are the tunneling times, the activationist temperature-independent tunneling times, shown here with these points, down 2 beta strands coupled in this azurin model system, logarithmic tunneling times on this scale, and linear distance on this scale, so you can see yourself, because it's linear on this logarithmic tunneling time scale, it's dead exponential. This series of experiments showed beyond any doubt that the mechanism of electron flow in the guidance system is superexchange coupling. And on top of that, it suggested that it's bond-mediated coupling, because the decay, 1.1 per angstrom, is very, very close to the sigma bond-mediated coupling that's been established both theoretically and experimentally. So we have electron flow that's mediated by bonds through this system, but our first system had covalent bonds. What about other bonds? What about other structural elements? How are we going to control the flow with bond elements? And we thought, well, van der Waals gaps must be inhibitory. Hydrogen bonds, we weren't sure. So we decided to map several other proteins that were much more heterogeneous, 4 helix bundle proteins like cytochrome B562. And I asked Ralph Langen to make 9 tunneling mutants and put probes in these positions shown here. In this heterogeneous system, we're tunneling through alpha helices in different directions, tunneling through hydrogen bonds, tunneling through gaps. And what I wanted to establish here is, what is the scatter? What is the bond scatter through this benchmark beta-strand tunneling time? And here's the scatter. The scatter, and then we mapped several proteins, and here's the scatter around the benchmark 1.1 angstrom tunneling time. There's big scatter in right in this region, big scatter. And the scatter, then theoreticians went to work and explained it all. It turns out that the hot spots here are regions where you tunnel through short hydrogen bonds. and the cold spots over here where you're forced to tunnel through van der Waals gaps, which are highly inhibitory. And so the bonds, the nature of the bonds in the proteins communicate and direct the flow of the proteins. We also mapped aqueous glasses to see where tunneling through water occurs and through vacuum. And so with this master sort of tunneling timetable through vacuum, through water, and through various types of proteins, we were able then, with the structurally determined cytochrome oxidase, to map out the guidance system through cytochrome oxidase to find out in these membrane proteins they're hardwired. They're hardwired with tunneling pathway through the system goes like this. You can tunnel through here in a few microseconds, tunnel across to the oxygen complex. The gaps are in here. You can't just get an electron just jumping in here to make a reactive oxygen species. It's hardwired, so as it goes through various conformational changes, the electrons can always be delivered to the oxygen to make water. It's a long story, but I don't have enough time to tell you the full story because I want to move on now to photosynthesis. If this were the only story, then photosynthesis would never work if it were only single-step tunneling, because the reaction would go the same in both directions. And it has to be faster, much faster in one direction than the other direction in order to store solar energy. And it goes like this. In photosystem 2, you excite P680, and now the tyrosine is critical, because P680 oxidizes the tyrosine, and the tyrosine oxidizes the manganese water oxidation cluster. Jim Barber will talk about this structure. I just have a cartoon up here. But the point is, in order to pump it out to store solar energy, you have to have hopping through tyrosines and tryptophans and things like that. So short hops, short tunneling, multi-step tunneling, which we call hopping, in one direction only. So the idea is you excite P680, you have two short tunneling times, which are very, very fast. So hopping through the tyrosine means that this reaction that goes in to put holes into the oxygen evolving complex so you can oxidize water to oxygen, this is over 100 to 1,000 times faster than the single-step tunneling back. So since it's differential hopping in this direction only, the system, because of the way the energetics are, it's forced to single-step tunnel back. So in this model, where hopping beats tunneling by quite a bit, you do differential hopping to store solar energy, and by the time the hole is in this oxygen evolving complex, the electron is all the way out to the quinone. It's now miles away. Otherwise, it would fall back to make heat, and we'd never store any solar energy. Now, being that far away, it takes centuries in single-step tunneling, because the power of an exponential, single-step tunneling back takes forever. So this allows, in hopping, it allows you to pump out the oxygen evolving complex 4 times without any electrons falling back into it to make heat. This hopping advantage, we decided we would check again in a model system to see if this is right. So we built another model system in Azure, where we have the copper here. We engineered a tryptophan in between as the hopping station, and we put a rhenium sensitizer here, because the excited state of this rhenium sensitizer has exactly the same potential as excited P680, 1.4 volts. We excite this rhenium, it oxidizes the tryptophan, and it hops then to oxidize the copper. And in this experiment, we showed directly by laser-triggered optical absorption that the hopping beats the tunneling. This is the hopping that oxidizes the copper in a few nanoseconds, 200 times faster than single-step tunneling. The single-step tunneling back reaction is here in microseconds. Hopping beats tunneling, a direct demonstration. We published this in Science. We worked out all the details through time-resolved infrared work, fluorescence, laser-triggered optical absorption. The bottom line is the hopping that goes through the tryptophan oxidizes the copper super fast, and the back reaction is slow. We published this in Science in 2008. And then we worked out a number of coupled differential equations and these kinds of hopping things so we could create hopping maps. The hopping maps we generated from information we had about coupling, superexchange coupling, and barriers to make a hopping map so we could predict exactly how much the hopping would beat tunneling, what the advantage would be. On the vertical axis, the driving force to get the hop. On the horizontal axis, the overall free energy driving force for the reaction. And we predicted in this first hopping map that we could predict the hopping advantage in this model system almost to within a factor of 2, a remarkable thing based on our fundamental ones. And then we figured if one tryptophan is good, 2 must be better. So we decided to build a tryptophan wire and a double hop in our system. This pushes the donor and the excited state further and further away. And we found in this experiment that the double hop through 2 tryptophans is just as fast as a single hop, but the back reaction, now being very, very far away, is now very slow. And now the hopping advantage is a full 10,000. And so as you build this thing out, the hopping advantage increases more and more and more, indicating how you can actually build systems to store solar energy. That is, the excited state reaction super fast, the back energy wasting reaction slower and slower as you build this system out. And with this hopping advantage, we made hopping maps of some of the interesting systems that people suspect hop, like photolyase and ribonucleotide reductase. And we showed in these hopping maps for these important enzymes that you have to hop. You can't do these reactions over these long distances, say in ribonucleotide reductase, without hopping through these tyrosines that in between the oxidizing site and the active site. Then we focused our attention on using this trick of generating holes deep into enzymes through hopping. We decided to work on cytochrome P450 and put our probes in that, put our probes in P450 and next to tryptophan, so we could flash quench these things and generate a hole deep into P450 to generate compounds 1 and compound 2, which Mariah Enner showed that we could do. But we noticed something very interesting in generating by hopping, by deeply pushing this hole into the enzyme. We found that the yield, the quantum yield for the compound 2 was very low. Only out of 100 photons, we only got three or four that were efficient. Something else is happening in these enzymes, these oxidative enzymes. The holes are going someplace else. We had this beautiful system with a sensitizer, a tryptophan deep into the active site of P450 to generate the active intermediates. But we only generated them very, very low efficient. And then we started looking at P450s and how efficient they are. And what we found was that the human P450s, the really big time ones that do steroid biosynthesis, the human one that oxygenates cholesterol, is only 10% efficient. It's very inefficient. So the holes must be going, there's someplace else the holes are going. And so we started looking for where these holes are going into P450. For example, only 10% of the reaction is going through the efficient hydroxylation cycle. 90% of it's going through this cycle that, if it weren't intercepted, would lead to decomposition and death, would lead to very rapid aging because of the generation of enormous number of reactive oxygen species. And so Jay Winkler and I propose that there is a protective system in all of these enzymes that utilize oxygen. And the protection system starts with the tryptophan. This is our hypothesis bank that we're putting in your QRB discovery. This is our new work. We hypothesize that there will be a tryptophan, maybe some tyrosines, that will actually intercept these holes if the system generates and the substrate's not there. The holes will be intercepted by tryptophan and maybe handed off to tyrosines to get out to the edge of the protein, where the system can be protected by reduction by glutathione. So we followed this up by starting to look for tryptophan and tyrosine chains in proteins. And what we found in BM3 immediately was a tryptophan-tyrosine mini chain that goes out to the surface, where it can be protected by glutathione. And then we've made survival maps to see how fast this could occur. And we found that's of order of microseconds to a few nanoseconds that you can get these holes that are generated you don't want, you can get them out to the surface very, very quickly so they can be reduced by glutathione. And then we started looking all through the protein data bank for all the enzymes, and we found their chains, their tyrosine tryptophan chains in all the oxidative enzymes. I'll just give you a couple just to finish this talk. Here's the chain that we discovered in the human P450, this one that oxidizes cholesterol. There's a long chain that starts with tryptophan and then goes through a bunch of tyrosines out to the edge. And we can calculate the survival time of a whole through this big chain that we've discovered in the structure of this enzyme, and the survival time is very short. So it's timed perfectly so that it's not faster than the actual turnover with cholesterol is there. That's actually faster with compound 1, a little faster. But if cholesterol isn't there, the whole can get out to the edge through this chain to be reduced by glutathione, so the enzyme is protected. So there's no reactive oxygen species. Even more impressive is the chain we found in methane monooxygenase. Several chains, huge chains of tyrosines and tryptophans. Here they are. There's the active site of methane monooxygenase. This is a big chain that goes way out to the surface. The tyrosines and tryptophans are only 3 to 4 angstroms apart. They're coupled together, so the single step, the tunneling through them is extremely fast. And it's dictated by the first step into the tryptophan. And that's timed exactly right so that you can get the turnover with methane to methanol in the enzyme. But if methane is not there, the system is protected by these systems. To finish the talk, so that I'm on time, Mr. Chairman. In fact, 5 minutes, I think, to go, or 10, or whatever it is, so that we can have discussion. We found these chains all through the protein data bank. We've looked at, we wrote some code to ask the question, are there tyrosine and tryptophan chains that are closer together than 5 angstroms? And what we found is they're all over the protein data bank of all the structures. Here's the whole story here of the chains, the 3, 4, 5 chains. Of course, in the enzymes that utilize oxygen, these chains are much more prevalent, much longer. There are ones up in 4 or 5 tryptophan. We think, and we're publishing in QRB Discovery Bank, this idea that these chains have been built into these oxidative enzymes for protection. With that, I'll thank my coworkers so we can have time for discussion. Jay Winkler, and tragically, we've lost this coworker in the last 2 weeks. Jack Richards passed away suddenly, and very sorry to report that. These two have been senior coworkers of mine for many years, and we just lost Jack suddenly. The students and postdocs, Mariah Inner, Kana Takamatsu, and Jeff Warren, these folks have supported me. Thank you very much for your attention.
Thank you. Thank you very much, Harry, for this wonderful talk. And at last, at this conference, we heard about some tunneling electrons. I really like that. So,
good.
Thank you very much. There are time for some questions.
Yeah, I left time for questions.
First question from Bengt Nordén.
Hello. Harry, I promised myself not to ask any questions, but I can't help myself now.
Okay.
Thanks for a fantastic talk.
Thank you.
You have sort of changed paradigms a number of times by your discoveries. I think many organic electronic people would say that conjugated bonds would be the way to effectively transfer electrons. But you have the two mechanisms. You have the super exchange, tunneling, even hydrogen bonding being ported.
We don't need conjugated pi systems except for tryptophans and tyrosine to do multi-step tunneling.
And then you have the hopping, the rectifying effect of hopping.
We can't use double bond systems or silicon or anything like that for the single-step tunneling because they're oxidatively unstable.
Yeah.
You have to have a very robust system in aerobic life. But this system that uses sigma bond tunneling for single-step tunneling and uses the other elements, and then for long-distance things and faster uses hopping, but still multi-step tunneling for each step through these sigma bond systems, that replaces the conjugated pi systems that would push electrons a long way just in single-step tunneling through pi bonds. But the robust system I've described replaces that. It's robust, it's oxidatively stable, whereas a series of double bonds all the way through would not be.
So let me ask the question now.
Okay.
What advice would you give the organic electronics people how to strategically use the knowledge, the counterintuitive discoveries that you have made in order to optimize development of transistors, diodes, and solar energy devices?
Don't use conjugated pi systems, use peptides. That's clearly the message. Use the power of biological systems and all of the things you can engineer in a protein or a peptide system to replace a sort of a wimpy organic pi framework, which isn't going to last very long in an aerobic planet. So my advice to the organic chemists is change to inorganic chemistry.
Thank you. So, next question.
Well, thank you for a very interesting lecture and good advice, I would say, as a biophysicist. Yeah. However, what about the oxygenases? Have you been able to trap any radicals on the way out there in any of your experiments yet?
That's exactly what we're doing now experimentally. We've started a program now that we have just discovered these new chains, and they're all over the place. Yes. So it's unbelievable how many of these chains, and they're all in these enzymes that use oxygen.
Yes.
Every enzyme that uses oxygen, we found a long chain that starts with tryptophan and then has a bunch of tyrosines. And by the way, the tyrosines aren't just normal tyrosines. They're all coupled to bases, so they're redox active.
Yes.
And so they look like, it just looks like the holes are going to go right through there fast to the surface. Now what we're doing is, at the moment, is we're attaching probes to the end of the last tyrosine. An osmium probe that has the potential to actually take the whole from the last tyrosine, and we get a big optical change there. And so we're now engineering these things at the end of these chains so we can trap these radicals and really see if our survival time calculations, which come from our hopping maps, whether that's right or not. Because we were predicting, as you saw, we're predicting microseconds to hundreds of nanoseconds for this whole transfer out to the surface. And so now experimentally, we've got to check that.
Yes.
And that's exactly what we're doing. But in Bink's journal, we've got some of these chains, and we're reporting. We think it's a hypothesis that these are protective chains.
Yeah. Great, thank you.
So another question.
Harry, obvious proteins to look at are photosystem 2 and cytochrome oxidase, which of course, metabolize oxygen in a way.
Yeah, cytochrome oxidase has a big chain like this.
Oh, it does?
Yes.
What about
absolutely.
photosystem 2 then?
It's got a big chain like this in case, you don't get the things over to the oxygen or the ferryl species. It's on the way to water. If it goes wrong, there's a chain that protects it. I haven't looked at photosystem 2 yet.
No. You would expect it there, wouldn't you, Ray?
Yeah,
if
sure.
Your hypothesis is correct.
I think we're going to find them in everything that has high potential intermediates that would lead to reactive oxygen species if you don't intercept them. Otherwise, we'd be aging like in 2 or 3 days.
Well, I think in photosystem 2, there are other ways in which you can quench high oxidation states through electron transfer from actual cofactors like the cytochrome and so on.
All I know is I've gotten as far as the oxygen ones, and cytochrome oxidase has a big chain.
Okay, so I think that's going to be the last question, and we're going to get back to some of these things, I think, in the panel discussions in the evening. So join me and thank Harry again for a wonderful talk.
Thank you.